Nature. Oh, how we love it. The song of the birds, the gentle breeze, the smell of rich earth and moss. But what does nature actually mean? What do we humans perceive as natural? Is chemistry part of nature, or even genetics? What we see here in front of us is the fruiting body of a fungus. It is composed of millions of connected fungal cells, the mycelium, which extends far and wide beneath the surface. Fungi are masters of recycling. They break down all biological material, convert it, and use this energy to grow and reproduce. Fungi are constantly cooperating and competing with other organisms like bacteria for nutrients and habitat. Here in the forest in particular, fungi are a fundamental part of the ecosystem and its cycles. Let's delve into the fascinating interior of a fungal cell. Welcome to the BiMM Research Station. We now find ourselves in a dimension that usually remains hidden from the human eye. The nano world. Here, distances are measured in nanometers. 1 million nanometers make up 1 millimeter. Here, for example, is a water molecule. It consists of 2 parts hydrogen and 1 part oxygen. On our scale, it is roughly the size of a watermelon. What surrounds us here is the cytoplasm, a dense sea of molecules that are constantly colliding and reacting with one another. Yet within this apparent chaos, there is also order and clear plans. These plans are stored in a secure central location, the cell nucleus. The nuclear pore complex is a strictly guarded gate in the membrane of the cell nucleus. It functions like a checkpoint with built-in gatekeepers who check very carefully which molecules are allowed in or out. Because here, inside the cell nucleus, lies something very special. The code of life, the DNA. These DNA strands contain a sequence of just 4 different nucleobases. They are abbreviated as A, T, G, and C. They form a kind of universal genetic language that forms the basis of all life on Earth. The order of these letters provides the blueprint for everything a cell can produce. Most of these building plans are used and read constantly, but some of them remain inactive for long periods until they are needed. One example are genes necessary for defense. They are only activated in case an enemy is around. Here we see the cell's workers, the ribosomes. They are located on the walls of the endoplasmic reticulum. The ribosomes read the gene's blueprints and assemble the proteins that ultimately form all cellular structures and make them function correctly. For all this to be possible, cells need energy. Here in the mitochondria, this energy is generated from sugar and oxygen. They act as the cell's power stations and are distributed throughout the cell's interior. The newly formed proteins from the endoplasmic reticulum are then packaged into vesicles. So-called motor molecules act as transporters, picking up vesicles and transporting them to the appropriate destination within the cell. There they are unpacked from the vesicles and eventually can perform their intended function. The tubular street networks, called microtubules, not only ensure traffic, but they also give the cell structure and stability. That is why they are part of the cytoskeleton. In case proteins need to be incorporated into the cell membrane or are destined to leave the cell, they are incorporated into a dedicated packaging system called the Golgi apparatus. There they additionally are decorated with a special sugar coating on their exterior, which ultimately makes them more stable and resilient in the potentially hostile external environment. The processes we have just observed in a sort of time-lapse view actually take place at lightning speed in a fraction of a second. The cell permanently and constantly reacts to signals and changes in its environment. It is precisely these processes that make fungi so interesting for research and development. Fungi are masters at producing chemical substances which are used by themselves for communication with their environment, regulation of developmental processes, or for defense. That is why researchers need to understand the processes within the cell down to the smallest detail. If, for example, the fungus is threatened by other organisms or lacks certain nutrients, the silent building plans in the DNA are activated. Simulating such conditions in the laboratory is extremely difficult. But with certain epigenetic tricks, these hidden treasures within fungal cells can be uncovered. Treasures that could potentially be of great value to medicine, agriculture, or biotechnology. The significance of this research becomes even clearer when one considers the diversity of fungi. Estimates suggest that there are up to 6 million species of fungi on Earth. So far, however, only around 150,000 of these have been scientifically described. An immense source of potentially new bioactive compounds and biological innovations still lies hidden. Science and research thrive on curiosity, dedication, and a willingness to explore new avenues. Nature, too, is a vast laboratory where experiments have been taking place for billions of years. Through the interplay of countless factors and coincidences, nature has produced a sheer, almost incomprehensible diversity. Complex, surprising, and full of secrets waiting to be unraveled.